the disorder, before disorders of fatty acid oxidation, we had to do regulation of beta oxidation. So let's start with the regulation. As you already know, regulation of beta oxidation, you can see, I have told you, first beta oxidation happens in fasting condition, and in fasting condition, you know, fasting and starvation condition, it happens, and under influence of glucagon. And in glucagon, what does this glucagon do? Glucagon in adipocyte, it breaks triglyceride into fatty acid and glycerol by having its influence on hormone sensitive lipase. So after it being broken, this triglyceride being broken down into fatty acid, and this fatty acid through blood, it will reach liver. It will reach liver and other peripheral tissue through blood, fatty acid, glycerol also. But let's talk about fatty acid. Now fatty acid, what will happen? This fatty acid will be activated by outer mitochondrial membrane. So fatty acyl coenzyme A, and this fatty acyl coenzyme A will go into the mitochondrial membrane, will enter mitochondrial membrane as fatty acyl carnitine, and then again this fatty acyl carnitine converts into fatty acyl coenzyme A. So this transport, here is transport and this. So there are two regulatory steps. First is because glucagon, you can see this is first regulatory step at adipocyte, and second regulatory step is at transport of this fatty acid through this mitochondrial membrane. Now you can see when this is when the condition switch to fat condition, what will happen? Insulin will be high. And then insulin will inhibit this enzyme. This insulin will inhibit this enzyme. So this lipolysis will also be inhibited at adipose tissue. And second, there will be production of from acetyl coenzyme A. There will be production of high amount of malonyl coenzyme A during fat condition. Malonyl coenzyme A. So this malonyl coenzyme A will inhibit an enzyme that is required for transporting this fatty acid through this mitochondria. So you can see this is how cell ensures fatty acid oxidation only during fasting condition, not during fat condition. Fat condition, there are two mechanisms that inhibits. First is insulin, and second is malonyl coenzyme A that inhibits fatty acid oxidation and promote fatty acid synthesis. So you can see here. Can you see this? Lipolysis and availability of free fatty acid will be ensured by glucagon, high glucagon and low insulin, by stimulating hormone sensitive lipase. And then that is how free fatty acid will increase in fasting condition. But opposite is insulin inhibits this enzyme during fat condition. That is why fat condition facilitates fatty acid synthesis. Fasting condition facilitates fatty acid oxidation. Now, second thing you can see, malonyl coenzyme A, that is synthesized from acetyl coenzyme A during fat condition by acetyl coenzyme A carboxylase, will inhibit the transport, this step, the transport of fatty acid into mitochondria. So these two are very important regulatory steps, one in adipocyte, other in transport of fatty acid. Two are very key steps. And then in this transport of fatty acid also, there is mitochondria outer membrane and inner membrane. This CPT is present in CPT-1 is present in outer membrane. So this will inhibit this enzyme. So malonyl coenzyme is present, inhibit the CPT-1 that is present in outer mitochondrial membrane. Okay, so that is how this transport of acyl coenzyme A or fatty acid is not allowed because of inhibition of CPT-1 in fat condition. So fat in condition, there will only be fatty acid synthesis, not oxidation. By these two mechanisms. And in fatty acid, in fasting condition, this activation of these two mechanisms will ensure fatty acid oxidation. Now, when there is high amount of fatty acid oxidation, you know there is product will inhibit the reaction. The product is NADH and ATP. Ultimately, the product will inhibit the fatty acid oxidation. And then reactant, this is reactant. Reactant will have stimulation effect on this whole fatty acid synthesis. High fatty acid, if there is high fatty acid, it will stimulate fatty acid oxidation. Product, that is NADH, FADH, will inhibit the reaction. This is feed forward stimulation, feedback inhibition. That will work here. Now, this is two. These two are very more important than this. Now you can see one very important regulation that is stimulated by your exercise. So exercise will stimulate. Exercise will also stimulate fatty acid oxidation. How? So exercise, what does it do? Exercise will stimulate this one type of kinase. This is called AMP kinase, because exercise reduces energy. Exercise increases AMP inside cell, high amount of AMP. Now this, because of this, in muscle cell and other tissue, this AMP kinase will be activated, and this AMP kinase actually inhibit this reaction, inhibit the formation of malonyl coenzyme A. If malonyl coenzyme is not there, so this will not be inhibited. And that is how the flow of fatty acid will be easy through the mitochondria. So exercise, you can see exercise stimulates fatty acid oxidation through AMP kinase. Remember, how does exercise stimulate fatty acid oxidation? Through AMP kinase. AMP kinase. AMP kinase. How does, so the question comes, how does exercise stimulate fatty acid oxidation? Oxidation. It is through AMP kinase. This. Did you understand this whole process of regulation of beta oxidation? No. Did you understand? Tell me.
Sir, please, I have a question.
Yes.
So how do people lose weight by exercising if exercise would stimulate AMP kinase?
So exercise, when exercise stimulates AMP kinase, what it will inhibit this enzyme. This is enzyme of fatty acid synthesis. Okay, and if it inhibits, this will be reduced. And you know this is an inhibitor of fatty acid oxidation. This, if this is not there, malonyl coenzyme is not there, what will happen? Fatty acid oxidation will be facilitated.
Okay, thank you, sir. I get it.
Okay. So this is the reason. Remember this. I remember this AMP kinase. AMP kinase is associated with exercise. Exercise function through this AMP kinase. This is very friend of exercises. AMP kinase is friend of exercise. I cannot tell you I cannot go into very detail of everything, but just remember, exercise means AMP kinase. That will help you later. Now, disorders. These are the enzymes you can see. Is there any problem? Anyone want to ask question? Okay. No. So we will talk about the disorders of fatty acid oxidation. So if we talk about disorders of fatty acid oxidation, so any enzyme that are involved in fatty acid oxidation, deficiency or defect of any enzyme can cause fatty acid oxidation. So these are the list of enzymes. Among these are those you see in very fed later. These are rare conditions. These are rare, not that frequent. These are rare. Though all of these are rare, and this is rarest, these two are commonly seen. If you see the patient, these two are more commonly found disorder. One disorder is CPT-2. CPT-2 is, what is CPT-2? Carnitine palmitoyl transferase. That you see, this is inner mitochondrial membrane and outer. CPT-1 is present in outer mitochondrial membrane. I told you it is a rate limiting enzyme that is inhibited by inhibited by malonyl coenzyme. And you know this, sorry. So this actually facilitates conversion of what? Acyl coenzyme A to acyl carnitine. This function of CPT is acyl carnitine. Now this acyl carnitine will enter into mitochondria through the protein CAT. And then this acyl carnitine has a carnitine will have to convert it again into acyl coenzyme A, and this is done by CPT-2. This acyl carnitine will convert into acyl coenzyme A by which enzyme? CPT-2. Now tell me one thing, if this is absent, what will happen in our body? So there will be high amount of this acyl carnitine in our body.
Yes, sir.
Yes, did you get this? So acyl carnitine will be high because CPT is not working, and that is why acyl carnitine will become high. Now second disorder you will see, this is because of MCAD deficiency. MCAD, what will happen? So medium chain fatty acid, I told you, from long chain fatty acid also it will ultimately convert into medium chain. So medium chain fatty acid, after it being entered into the tissue, so medium chain fatty acid is generally 8 to 12, C8 to C12. So you can, I hope you can understand this. This acyl coenzyme A, this 8 to 7 acyl coenzyme A will not be catalyzed to acetyl coenzyme A. Will not be catalyzed to acetyl coenzyme A. And as a result, what happens is this coenzyme A, when it is very high inside cell, it can go back outside the cell, and then what happens is C8 to C6, C12, this type of fatty acid coenzyme A can convert into C8 to C12 carnitine. Did you get that? So this, in this condition, you will see the patient have very high amount of C8 to C12 medium chain acyl carnitine. You can call it medium chain acyl carnitine. Acyl carnitine. So what you see here is very important concept. Laboratory parameter, when you have to test this patient, you have to test for acyl carnitine. Acyl carnitine. If acyl carnitine is very high, the patient has disorders of fatty acid oxidation. So any patient, any baby, actually newborn baby, we test this in newborn baby, because these are all disorders of these are all called inborn error of metabolism. Error of metabolism. So see, in biochemistry we will talk about inborn errors of metabolism. So this is the first class you have. First class actually I already done in carbohydrate, Hurler and Hunter. It is also inborn error of metabolism. This is maybe second class of inborn error of metabolism. So inborn error of metabolism. These are all inborn error of metabolism. Remember this. We place all this disorder under inborn error of metabolism. So this inborn error of metabolism, all the inborn error of metabolism, we test baby after baby is born through this test, acyl carnitine. We send blood, very small amount of blood is taken, and then we ask for this test, acyl carnitine, from laboratory. If acyl carnitine level is very high, we suspect a baby having disorders of fatty acid oxidation. Did you get the significance of this whole concept? Did you get?
Yes, sir.
Okay. So I told you acyl carnitine. I also told you CPT-2 and CPT. We will talk in detail about CPT-2 and MCAD deficiency. No, we will take a overview of fatty acid oxidation disorder. So you know, fatty acid is required. What is the function, main function of fatty acid? What is the main function of fatty acid? It is energy during fasting condition. It is a fuel. Fatty acid is fuel. It is fuel of fasting and starvation. Do you agree with me?
Yes, sir.
Now, so this fuel, if fuel is not there, if fuel is there but it is not metabolized. So if fuel is not metabolized, what will happen? Energy will be low. Energy will be low. This is the center of pathogenesis of this disorder. Center of. So patient will have low energy during fasting condition. Patient has intolerance to fasting. The baby will have intolerance to fasting. And this baby, which tissue is involved? The tissue, peripheral tissues are involved very much, and also central nervous system. Tissue involved, high energy requiring tissue will be involved, because when there is no fatty acid oxidation, all tissue try to oxidize carbohydrate. Then ultimately brain will also have deficiency of carbohydrate, because the carbohydrate that is supposed to be used by brain will be utilized by other peripheral tissue. And that is what brain will also have a sort of a scarcity of this fuel. And this fuel is not there. Even though it is there, it is not being oxidized. So energy will not be produced from this. So all tissue, especially muscle tissue, will be involved. Almost all tissue of the body will be involved. Muscle tissue, which muscle? Cardiac muscle. Cardiac muscle. So cardiac muscle will be involved. Cardiomyopathy will happen. Now skeletal muscle will involve. Skeletal muscle. Other tissues, liver will be involved. Liver, brain will be involved, and then eyes will be involved. These all multi-organ will be involved. And what happens is, the very deadly condition is sudden death without warning. This is the very peculiar feature of this disorder, sudden death, because of lack of energy. Sudden death happens in such patient. Without any warning, sudden death happens. This is the very peculiar feature of fatty acid oxidation disorder. So did you understand the pathogenesis? Energy is the center. Lack of energy is the center. Why? Because beta oxidation is not functioning properly. Okay? So you can see this is CAT, this is carnitine CPT-1, VCAD, VLCAD, LCAD, MCAD, SCAD, and this trifunctional protein. This all can be deficient. Any one of these can be deficient, but the most common is this. We will talk about this most common. CPT-2 is associated with muscle. Predominant presentation will be muscle pathogenesis. Just remember, CPT-2 means muscle, and MCAD will be associated with liver. So we will talk about these two disorders. Now, these are general presentation, as I have told you. So you can see heart will be involved, cardiomegaly, other nervous system is involved, muscle is involved, liver is involved, eye is involved, kidney is involved, and this congenital anomalies will be happen. And maternal, if mother has such symptom, the mother will have a different problem. I'm not going to talk in detail about this. Okay, so I will be focused on these two, liver and muscle pathology. Liver is related to MCAD deficiency, liver pathology, and this muscle is related to CPT-2. So muscle damage will cause myoglobin in urine. This is called myoglobinuria. Myoglobinuria is the, you can see this is myoglobinuria. You can see urine is like dark color urine, and this is because of damage to muscle, and muscle damage is called rhabdomyolysis. Skeletal muscle damage is called rhabdomyolysis. This, in such condition, patient will not be able to do exercise. So here you will find exercise intolerance. In such condition, you will see fasting intolerance. So there are liver pathology present with fasting intolerance, and muscle pathology present with exercise intolerance. See the difference. Both intolerance happens both places, but one is exercise, other is fasting intolerance. So remember these two presentation, and this is a very important terminology. Hypoketotic hypoglycemia, or also called non-ketotic hypoglycemia. Hypoglycemia, because hypoglycemia is related to fasting intolerance. If hypoglycemia happens during fasting condition, fasting cannot be tolerated. And this fasting is what type of this hypoglycemia is related with absence ketone. Ketone body is not there. Generally during fasting condition, ketone body rises, but there is no ketone body because fatty acid oxidation is affected. And you know the source of ketone body is fatty acid. That is why non-ketotic hypoglycemia will be seen in MCAD deficiency and CPT-2 deficiency, exercise intolerance, and rhabdomyolysis. Now, will you remember? Just tell me. Do you remember? Anyone tell me what happens in MCAD deficiency?
Hypoketotic hypoglycemia.
Okay, hypoketotic hypoglycemia, and this means fasting intolerance. And then CPT-2, carnitine palmitoyl transferase 2 deficiency, will lead to what? Muscle pathology. What is it?
Exercise intolerance.
Rhabdomyolysis means lysis or damage to skeletal muscle. This leads to myoglobinuria, because myoglobin is present in muscle. It stores oxygen for the muscle. Myoglobinuria. And this actually, why does it happen? When you do exercise. And this is called exercise intolerance. Exercise intolerance. So did you understand these two conditions? Will you be able to separate these two conditions? So in all this condition, you have, as I have told you, this acyl carnitine is in blood is high, and we test newborn. This test we test newborn for acyl carnitine, high acyl carnitine. So now let's see this. So these two disorder I have already described you. What happens when it's MCAD deficiency, medium chain fatty acid coenzyme dehydrogenase deficiency? So pathogenesis presentation will be related to liver. Here presentation will be related to muscle, CPT-2. It is easy to, there is no confusion. You will not have confusion. I told you. You can see here in such condition, the baby will have C8 to C10. I have told you C12, no problem. C8 to C10. That medium chain acyl carnitine will be high in blood of baby, newborn baby. So this is the condition you will see MCAD deficiency. What is there? So fasting hypoglycemia. Hypoglycemia is there, and there will be absent or low ketone. Ketone will be low or absent. So that is why non-ketotic hypoglycemia or hypoketotic hypoglycemia. Both hypoketotic hypoglycemia or non-ketotic hypoglycemia. Both terminology can be used. Don't confuse. Both are same, related to same disorder. You can see the infant episodes provoked by overnight fasting. So if infant is goes for overnight fasting, then the infant will feel like sluggishness, and then infant will feel comatose. Infant will have difficulty in feeding because of lack of energy. Now overnight fasting and other conditions are disease. Some illnesses can provoke this, and then that causes loss of appetite and vomiting. That these disorders can cause, or overnight fasting can induce this hypoglycemia, non-ketotic hypoglycemia. So what is the cause of this hypoglycemia? Why is there hypoglycemia? Non-ketone body, you understood why is there ketone body. It is not that difficult to understand. Why is there not ketone body? Because fatty acid is not metabolized. That is why no ketone body, because fatty acid ketone body is produced from fatty acid oxidation. So it is understood. then why is there hypoglycemia? Did you understand why non-ketotic? First tell me. Did you all understand why there is no ketone body or low ketone body?
Yes.
Because ketone body is derived from oxidation of fatty acid, from acetyl coenzyme. Ketone body is synthesized from acetyl coenzyme. That is why. Now, why hypoglycemia? Because, see, let me tell you one thing. So during fed condition, no problem. We have glucose from our diet. So during fasting condition, after, let's say, 18 hours. Okay, not after like 4 hours. Fed condition lasts 1 to 4 hours. Okay, fed condition is 1 to 4 hour fed condition. Then 4 to 18 hours. Or in round figure, you can take 24 hours so that you'll remember 1 day. So 4 to 1 day is fasting condition. Here, glycogen will supply the glucose, stored glycogen. But baby has very less glycogen. And then after fasting, there will be starvation condition. Starvation condition. After this period, there is a process called gluconeogenesis. Gluconeogenesis will maintain blood glucose. So this process will maintain blood glucose. Did you understand this? During 4 hours, dietary glucose is there. Between 4 to 18 hours, what is there? Glycogen is there to maintain blood glucose level. After 24 hours, 18 hours, or 1 day, you can say, which will provide glucose?
Gluconeogenesis.
Gluconeogenesis. And actually for baby, this glycogen storage is very less. This is for adult. So what will happen for baby? This gluconeogenesis becomes very important. This gluconeogenesis becomes very important for maintenance of blood glucose. Now, so let me tell you, what is gluconeogenesis? Just tell me. What is gluconeogenesis? So gluconeogenesis is synthesis of glucose from amino acids. Did you understand this?
Yes, sir.
Our body synthesizes glucose from amino acid and also from glycerol. Glycerol, that is synthesized during fed condition. Glycerol 3-phosphate. So glycerol and amino acid, our body form glucose. And synthesis of glucose is an endergonic process, uphill process. It requires one glucose, I have already mentioned, one glucose molecule synthesis require about 11 ATPs. If you can remember, do you remember this? If you don't remember, just understand this. So synthesis of glucose require ATP. Where does this ATP come during fasting condition? This ATP comes from beta-oxidation. So if there is no beta-oxidation, do you expect this ATP?
No.
Do you expect this glucose?
No.
So what is the result? Hypoglycemia. Hypoglycemia because of this fatty acid oxidation defect. So did you understand the concept? Why is there hypoglycemia?
Yes, sir.
So, and this beta oxidation is not there. So acetyl coenzyme will also be not there, and that is why no ketone body. But if everything is normal, then there will be production of glucose also, there will be production of ketone body. So if there is problem in this beta oxidation, there will be non-ketotic hypoglycemia. And there are other causes of hypoglycemia as well, but there will not be low ketone body. So this ketone body level is very important to differentiate type of hypoglycemia. What is the cause of hypoglycemia? So if ketone body is very less, the cause of hypoglycemia is MCAD deficiency. So you can see this from this picture. So you can see amino acid will form glucose if energy is utilized. So this process is called gluconeogenesis. This is very important concept. So you have to remember this. MCAD, presentation of MCAD is this, and this is very important. Non-ketotic hypoglycemia. Understand this. Now what will happen is, so from protein amino acid you get, and then so from beta oxidation you will get ATP. So combined effect will be glucose during prolonged fasting condition. But what will happen is there is no beta oxidation of fatty acid. So that is why no glucose. No glucose means hypoglycemia. And then so no beta oxidation means hypoketotic or non-ketotic hypoglycemia. These are ketone bodies. Beta hydroxybutyrate acetoacetate, 3 ketone bodies are there in our world. No ketone body. So hypoketotic hypoglycemia. And you can see this also contribute for, because I have told you this pyruvate carboxylase is an enzyme. If you can remember it, pyruvate carboxylase convert pyruvate into oxaloacetate. And oxaloacetate is very important for this gluconeogenesis also. We will talk about this gluconeogenesis. And this is stimulator of this enzyme is acetyl coenzyme. And that acetyl coenzyme is derived from this. So this enzyme will also not be activated. And that is why this also contributes more to this hypoglycemia. There are one cause, just remember this cause, and this low activity of pyruvate carboxylase also contribute to hypoglycemia. Low activity of pyruvate carboxylase will also contribute to hypoglycemia. Did you all understand?
Yes, sir.
Okay. Now you have understood this very important concept. So non-ketotic hypoglycemia, see, this is a very important concept. Now, so just solve this question. 3-month-old baby presented with poor feeding, even loss of consciousness when in interval of fasting is more than 2 hours. On blood examination, baby has low glucose, no ketone body, and C8 acyl carnitine. So what is the diagnosis you will make?
MCAD?
Deficiency.
Yes, hypoketotic hypoglycemia and C8, high C8 is there. So that is why MCAD deficiency, medium chain acyl coenzyme dehydrogenase deficiency. And then what is the pathogenesis of this defect? I have shown you the pathogenesis. This is the pathogenesis. Will you remember this? So this is how I will ask question. I will change this question a little bit, but the punchline, actually the question, the part of the important part of the question will always be this. These are the key points of the question. From this you will diagnose. So this is, you will see the consciousness, lack of consciousness when overnight fasting because of this infection. These are also contributing, supporting evidences. So question will be like this. Now other disorder you can see, I have already told you, rhabdomyolysis and myoglobinuria. Because there is no fatty acid oxidation, lack of energy. And a lack of energy will cause muscle ache. Muscle ache, and then when the patient starts doing exercise, try to do exercise, there will be exercise intolerance and damage to muscle and myoglobinuria. So these three are very key feature of CPT-2 deficiency. Okay, this is muscle specific deficiency. Generally, this is CPT-2 is present elsewhere also, but muscle specific CPT-2 deficiency is most common. So you will see such type of, and you can see the carnitine muscle somewhat similar syndrome. CPT-2 can be produced by muscle carnitine. Now muscle here also, in this condition also acyl carnitine will be high. So see this problem, okay? And treatment is give continuous glucose for both condition. Give continuous glucose diet and low fat, both condition. Low fat and increase carbohydrate diet as a treatment. And don't do prolonged exercise, prolonged exercise. These are the treatment. These are the treatment. IV glucose, just the patient comes, as soon as patient comes, just give the IV treatment. Prevention is frequent carbohydrate diet, low fat diet, and then avoid fatty meal and avoid severe exercise, okay, fasting, exercise, and infection. So these are the treatment. So must patients lead reasonable lives. So if you diagnose this patient early, the patient will not have problem throughout their life. If they know the problem, they know that they cannot metabolize fat, so they don't take fat. They only take essential fatty acids from fat, and then the rest of the diet. In rest of the diet, they include carbohydrate. Then they can survive easily. So the prognosis is not that bad. It is good. Patient can lead a normal life. Now let's talk a little bit about these peroxisomal disorders. So peroxisome, I told you the minor pathways happens in peroxisome. In every cell there is 100 to 1000 peroxisome. Do you know, in every cell there is 100 to 1000 peroxisome, and that is why if there is, and in brain cell the peroxisome function is very important for brain cell also, neuronal cell also, and other cell also. So you can see this peroxisomal disorder can be categorized into two groups. One group is enzyme deficiency. If there is enzyme deficiency, that can cause disorders that is related to peroxisome. If peroxisomal enzyme is deficient, one enzyme is deficient. Now, in such condition, only particular pathway will be affected. But this second type of disorder is called peroxisomal biogenesis disorder. There are certain genes called pexin genes, called PEX. So this gene synthesizes protein, and this protein are responsible for assembly of this peroxisome, and this peroxisome are assembled because of this gene. And if this gene are absent, this protein is defective, what will happen? Peroxisomal assembly will not be there. And if peroxisomal assembly is not there, what will happen? So there will be generalized defect of metabolism that is related to peroxisome. So these are called peroxisomal biogenesis disorder. We will talk about individual one by one. Refsum disease first we will talk, then XALD. This is called X-linked this is adrenal leukodystrophy. We will talk about X-linked adrenal leukodystrophy, and then Zellweger spectrum syndrome. Let's see very quickly. So what are the function of peroxisome? These are the function of peroxisome. You can see peroxisome, as I have told you, it is very important for beta-oxidation of very long chain fatty acid and alpha-oxidation. Okay, beta-oxidation of very long chain fatty acid and alpha-oxidation, fatty acid oxidation. Then amino acid catabolism. Also in amino acid catabolism also peroxisome is involved. You can see. So lysine catabolism, lysine papacolonic acid pathway. So for this catabolism, lysine has two pathways for its catabolism. This is one of the pathway. This pathway involves peroxisome, and in such condition, this molecule will accumulate if peroxisome is defective. Then hydroxyproline catabolism, and then this DS amino acid catabolism. These are the amino acid DS particles. Generally, our protein has L amino acid, all protein. But neurotransmitter, our brain neurotransmitter has got D-aspartic acid and D-glutamic acid. And catabolism, if there is no catabolism of this amino acid, the brain will have problem. So spermine is polyamine. This is very important during embryonic phase. So that is why there will be problem. Majority of their problem also arises because of failure of embryogenesis, because peroxisome is not functional. Now you can see synthetic. These are the synthetic function. Cholesterol, few reaction in cholesterol synthesis is contributed by peroxisome. Many reactions in bile synthesis is contributed by peroxisome. Plasmalogen. There is plasmalogen is a type of lipid. I did not tell you. This is triglyceride. But in triglyceride, first bond is ether bond. Fatty acid will be there. This is not fatty acid, this is alcohol. And this is fatty, second is fatty acid, this is phosphate. So this is plasmalogen. Ether, this is ether phospholipid. So plasmalogen is present in very high concentration in brain. So this plasmalogen synthesis failure can cause damage to brain. And then you can see this DHA. I've told you this is a very important fatty acid for development of brain. Docosahexaene 22 carbon 6 double bond containing fatty acid synthesis. So these are the synthetic function of peroxisome. You can see fatty acid oxidation, amino acid catabolism, and some synthetic function. For this all function, peroxisome is very important. So peroxisome damage can cause brain problem, CN central nervous system problem, not only central, peripheral problem also, kidney and liver problem. We will see that. So see, first let's talk about this Refsum disease. What is this Refsum disease? Refsum disease is called hypertrophic demyelinating sensory neural neuropathy. So it is a neuropathy. Peripheral neuropathy will be there. So anosmia, ataxia, retinitis pigmentosa, increased CSF protein. CSF protein will be very high. What is the cause? Let's first talk about what is cause. Cause is defect in alpha-oxidation. Alpha-oxidation. Alpha-oxidation happens where? Inside peroxisome. So there is one enzyme. The name of that enzyme is called phytanic acid. Phytanic acid. Alpha hydroxylase. This is a very particular type of fatty acid. Phytanic acid. This is a branch chain. This is a branch chain. Branch chain fatty acid. Like this is fatty acid. There is branching. So this is a branch chain fatty acid. And this fatty acid is present in dairy product, in ruminant meat like beef, goat meat, mutton, lamb meat. So this present in beef, lamb meat and other ruminant meat, in dairy product. It can be present in fish and greeny leafy vegetables, because in ruminant also, because ruminant means goat, cows, these animals eat grass. From grass, these are produced. These fatty acids are produced because of bacterial action inside the rumen of this animal. And this compound, phytanic acid, is metabolized in peroxisome by enzyme phytanic acid or phytanyl coenzyme A, alpha hydroxylase. If this enzyme is absent, what will happen? This branch chain fatty acid will accumulate, and that will be incorporated into nerve tissue, and because of that, what it will cause? Hypertrophic demyelinating. So nerve will try to heal, and this healing response is referred by hypertrophic nerve. Hypertrophic demyelinating sensory neuronal, sensory motor and sensory, both neuropathy. With anosmia, lack of sensation, smell sensation, ataxia, lack of coordination, and then retinitis pigmentosa, there will be loss of rod cells of the eye, and in CSF there will be high protein, because this blood brain barrier are breached, this nerve are damaged. That is why there will be high CSF protein. So these are the presentation of Refsum disease. Let's first talk about alpha oxidation. What is alpha oxidation? This is phytanic acid. You can see this is a 20 carbon fatty acid, branch chain fatty acid. If you count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, and there are 4 side chain, 16, 17, 18, 19 and 20. So 20 carbon containing this branch chain fatty acid that is present in dairy product or this ruminant meat, beef, lamb meat, goat meat, this all meat have this product. So what does this very fatty acid is metabolized in peroxisome by enzyme? This enzyme is called phytanyl coenzyme A, alpha hydroxylase. So what is alpha position? This is functional group. This is alpha carbon. This carbon is alpha carbon. So this very fatty acid will cause hydroxylation of this carbon. Okay, our mitochondria does not have this capacity. So hydroxylation, because of hydroxylation, this fatty acid will undergo its catabolism. So because of catabolism of this formal coenzyme A will be produced, the first component. Then this pristanic acid will form, formal coenzyme pristanic. Now this peroxisomal beta oxidation, this will undergo peroxisomal beta oxidation, peroxisomal. And this peroxisomal beta oxidation, what is beta oxidation, will release this bond. You can see this is alpha carbon, this is beta. So alpha carbon, this will be lost. Now again you can see this will form, this will CO group here, alpha beta. So bond will be lost here, and again like this here, and then bond will be lost here, and then you can see it will be lost here, and this bond will be broken, and you know the energy will be released because of that bond. Now you can see this bond lost because of peroxisomal beta oxidation, and peroxisomal beta oxidation will release this bond loss. Again peroxisomal beta oxidation, peroxisomal beta oxidation. So did you see how many compounds has been synthesized right now? Can you see? This is acetyl coenzyme A, this is 3 acetyl coenzyme A, 3 propionyl coenzyme A like this, and 3 isobutyl coenzyme A. 1 isobutyl coenzyme A. 3 plus 3, 1 isobutyl coenzyme A, 1 formal coenzyme A. So this is the catabolism of this phytanic acid. So first enzyme that is required for catabolism of phytanic acid is this. If this is absent, this phytanic acid will not be metabolized. This phytanic acid will not be metabolized. And if it is not catabolized, what will happen? It will accumulate and it cause Refsum disease. So that is what happens in Refsum disease. So Refsum disease, you can see Refsum disease is associated with peripheral neuropathy, retinitis pigmentosa, ataxia cerebral, and elevated plasma protein. You can see these are the problem. You can see the patient retinitis pigmentosa, lack of rod cell, and blindness. It will cause, this is hypertrophic demyelination. This is histological section. You can see in patient later stage, patient will have this type of scaling of skin. 4th toe is small. So this is also one feature. And you can see the patient will have also a heart problem because of this neuropathy of autonomic nervous system. The patient can have heart problem. So patient can have this cardiac problem, cardiac conduction abnormality because of problem in autonomic nervous system and nerve and conduction system in the heart. So this is Refsum disease. So remember this very important findings, and this 4 tetrad. This is very peculiar to Refsum disease. Peripheral neuropathy, eye, neuropathy, ataxia, and then this. You can remember like Refsum is R and RAN. RAN means R for retinitis pigmentosa, A for ataxia, N for neuropathy. So you can remember at least trial, and this peripheral CSF elevation you can understand from this. Now let's see about this disorder. The second, leukodystrophy is also because of single gene defect. X-linked, this is X-linked adrenal leukodystrophy. What does it mean? Adrenal gland is leukodystrophy means, what does leukodystrophy means? Leuko means white, dystrophy means loss. So loss of white matter. So this is actually loss of white matter. White matter. What is present in white matter? What forms white matter of the brain? Does anybody know? Myelin forms white matter of the brain. Did you understand? Did you understand this? Yes, this leukodystrophy means lack of myelin. And this lack of myelin happens because of this protein. This is a channel. of that is present in the peroxisome. This is called ABCD1, ATP binding cassette D1. Actually, this is responsible for transport of very long chain fatty acid to the peroxisome. Now, if this protein is absent, this protein is coded on X chromosome. If this protein is absent, the very long chain fatty acid will be very high in the blood. And because of accumulation of very long chain fatty acid, this will cause adrenal dystrophy. Adrenal cortex, you know, adrenal cortex has 3 hormones: glucocorticoid or corticoid, cortisol, and second is mineralocorticoid, and third is sex corticoid. So mineral imbalance in such patient, mineral imbalance will be there. There will be difficult to bear stress because glucocorticoid help in bearing stress. So stress bearing condition will be difficult stress bearing. The patient blood pressure can become low because of mineral imbalance and because of glucocorticoid deficiency. Patient can go into shock suddenly if patient encounters high temperature, low temperature, other stress condition. So this is the presentation of adrenal dysplasia. This is because of adrenal failure. And adrenal failure and leukodystrophy. There is no white matter in brain. So this is X-linked leukodystrophy. And it happens because of absence of this protein. And it is encoded on this X chromosome. And these are other type of leukodystrophy. This is sphingolipidosis. This one is called metachromatic leukodystrophy. Second is called globoid leukodystrophy. We'll study these 2 type of leukodystrophy in sphingolipidosis after we finish this fatty acid oxidation disorder. Now in next class we will talk about this